I hope that you're doing well. So we are going to talk about neurons, glial cells, and synapses today um, for our neuroanatomy class. So types of cell. Neurons are nerve cells. They generate and transmit information to control body functions, allow for thoughts, emotions, and memories. Glial cells surround and support neurons. Myelin is an example of a glial cell, and it helps increase the nerve conduction velocity. Neurons in the peripheral nervous system, or PNS, can heal or regenerate, versus neurons in the central nervous system, once they're damaged, they are irreversible. They die and cannot regenerate. The structure of a neuron, there is a cell body which contains the nucleus, the ribosomes, and the Golgi body. They're in the nucleus. It's the DNA of the cell is held in the nucleus. The ribosomes, they produce proteins, and those proteins are essential to the neuron's structure and function. The Golgi body processes and exports those proteins. If the cell body is damaged, the whole nerve dies. So if the axon is damaged and it's in the peripheral nervous system, it can be regenerated. In the central, it still dies. But if the cell body is damaged, whether it's central or peripheral nervous system, that whole neuron dies. Dendrites are what receive the information from another neuron. Axons pass along that information. The synapse relays the information to the next neuron. The synapse is also called the axon terminal, synaptic terminal, or the synaptic knob. So don't be confused when you hear those terms. And there are microtubules in the neuron that are like little motors that move substances around within the neurons. Ions, before we go to ions, mitochondria is the cell's powerhouse. The cell has a semi-permeable membrane. What that means is it allows some particles to freely flow through the membrane to go in and out, such as oxygen and carbon dioxide. They can freely go in and out of the cell membrane. But other particles, such as ions, must be carried through carrier proteins. They must be pumped in and or out of the cell. So ions are your charged particles. They must be pumped in or out of the cell. Ion channels are specific about what type of ions they allow to pass through. And one example of an ion channel is the sodium-potassium pump, which we'll talk about more. Ions move from an area of higher concentration to lower concentration. So if there's more sodium on one side of the cell, it naturally wants to go to the other side of cell. Positively charged ions are attracted to negatively charged ions, and the movement of these charged particles is called an electrical current. This is really important when we learn about like electrical stimulation for your modalities course. So it's good to have this foundational knowledge. Membrane pumps are required to move ions against the concentration gradient. So rather than what they naturally want to do, move from an area of high concentration to low, these pumps actually push them to an area where they're already more concentrated. This system requires energy in the form of ATP in order to move those particles against the gradient. Sodium-potassium pumps pump potassium into cells and sodium out of cells. I just realized it said to make the cell more negative inside than outside, which causes an action potential, which we will talk about later. A resting potential, this is the resting state of a neuron or a cell, nerve cell. So some ion channels are non-gated, and they continuously allow ions to flow based on their charge and the concentration gradient. So in the non-gated areas, they allow certain particles to freely go wherever they naturally want to go. The gated ion channels open in response to a stimulus, and otherwise they remain closed. And when they are open, they allow specific ions to pass through. But there has to be a stimulus that causes them to open. So at rest, the inside of the neuron has a negative charge. The reason it has a negative charge is because the sodium potassium pumps are continuously pumping sodium into the cell and potassium out of the cell. However, um, the non-gated ion channels allow the calcium to flow back out of the cell, but don't allow any movement of the potassium. So the potassium is being pumped into the cell, the sodium is being pumped out, and the potassium is being allowed to freely just move across that cell membrane through those non-gated ion channels to go back out of the cell. So as potassium is being pumped in, then it becomes an area of higher concentration, so it just naturally flows back out. And then sodium is being pushed out to an area of higher concentration. So then the outside of the cell becomes more positive than the inside. So the relatively negative charge is called a resting potential. This is the resting state of the cell, and the charge itself is negative 70 millivolts. Making sure there wasn't anything else I needed to say there. An action potential is a nerve impulse. So this is where the movement happens. So movement of ions creates an electrical signal that transmits the information from one neuron to another and eventually creates either a muscle contraction or a interpretation by the brain of maybe a sensation or something like that. So if the local potential of a neuron reaches a threshold of negative 55, then the sodium channels open. So once the inside of the cell becomes less negative because of an action potential, no, to create an action potential, the sodium channels open and the sodium rushes into the cell. This causes the inside of the cell to become positive or up to 35 millivolts, and that's called depolarization. So the inside of the cell, because the sodium has rushed in, once the sodium channels are opened, it depolarizes, which causes an action potential. So the neuron fires and moves the impulse along the axon to the next neuron. So it must reach the resting potential before it fires again. This is called a refractory period, which is basically a recovery period. So if a nerve impulse is passed along and it is still at that more positive or less negative environment, it's not reached its resting state, and another impulse were to come, then it cannot cause an immediate action potential to that area. So it causes a um, recovery period. So once the action potential occurs, then repolarization must occur to return that neuron to its resting state. So with an action potential, there's a resting state. Once the area becomes less negative and reaches a threshold, sodium channels open and sodium rushes into the cell, causing it to be more positive, which is depolarization. And once the nerve impulse is passed through, then there is a period of recovery where repolarization is happening to return it to its resting state. Um, this is an all or nothing principle where either an action potential happens or it doesn't. So it reaches the threshold and happens, or it doesn't reach the threshold and does not happen. What's really cool is once one area of the axon is depolarized, it forces the next sodium channels to open, so it immediately depolarizes too. So that creates a wave of depolarization. When one area depolarizes and it immediately opens the next sodium channels and the next open, and it just creates kind of a domino effect to create a wave of depolarization. Saltatory conduction. So myelin wraps around axons. It allows the nerve signal to skip over myelinated portions and jump from node to node. So in between the Schwann cell, which Schwann cells are in the peripheral nervous system and not the central, but there is myelin in the central as well. It's just not called Schwann cells. Um, but um, when the myelin is wrapped around, in between those groups of myelin are the nodes of Ranvier, and the impulse can jump from one node to another to increase the velocity of the contraction, the speed of the contraction. And that impulse jumping instead of just slowly sliding through is called saltatory conduction. So the movement of a nerve impulse by jumping from node to node is saltatory conduction. This speeds up the transmission of nerve impulses. Glial cells are those supporting cells. So the neurons are the main nerve cells. The glial cells are the supporting cells. So their function is to maintain the fluid around the neurons, to clean up debris, and to form that myelin sheath. So that increases the speed of nerve conduction. The types of glial cells, there are 5 different types. 4 of these types are all found in the central nervous system versus the last one, the Schwann cells, are only in the peripheral nervous system. So the first 4 I talk about are all in the central nervous system. So astrocytes, that forms that blood-brain barrier that keeps certain substances, such as certain drugs or certain toxins, from entering the brain. The oligodendrocytes, that is what forms the myelin of the central nervous system versus the Schwann cells in the peripheral nervous system. So oligodendrocytes form the myelin in the brain and spinal cord. The microglia, those remove dead or damaged cells and promote healing. The ependymal, might be saying it wrong, produces this cerebrospinal fluid and helps with circulation. And then the Schwann cells are what create the myelin in the peripheral nervous system. They also help with regeneration after injury. So they release a growth factor that stimulates the axon to regenerate if it's been damaged. But they also like create basically like a mold or a blueprint to help the axon find its pathway. Like this is where you go. There's already that mold or that blueprint there that it just follows along. It just fills up the space versus not being able to regenerate. So with the Schwann cells, if they are damaged, like with certain diseases like multiple sclerosis, then it could cause that nerve not to be able to regenerate. Okay, we already talked about blood brain. Okay, just making sure I was saying everything in the notes before I move on. Okay, um, with neuron repair. So once a neuron is damaged, which could be because of disease or trauma, or anytime there's a lack of oxygen to an area, it's going to be damaged. Um, then it may or may not be able to regenerate. If the cell body is damaged, the nerve dies, can't regenerate. If the nerve dies, it releases neurotoxins, which are chemicals that are toxic to nearby nerves and causes more neurons to die. So that's not a great process. I don't know why it does that, but I don't like it. So when a neuron dies, it releases neurotoxins, causing more neurons in that area to die. Um, if an axon is damaged in the peripheral nervous system, it may be able to regenerate, The way it regenerates, the Schwann cells produce that growth factor, stimulate the axon regrowth. The axons sprout from the intact cell body and go down until they reach a synapse. They grow very slowly, and if it's a long axon or a long neuron, long nerve, then it could take months or even years to fully regenerate and reach that synapse. But as long as it hasn't reached it, it will continue to grow. It must connect to another nerve to survive. So if it gets to a place where it just never reaches another neuron, then it will die. If the axons are damaged in the central nervous system, they cannot regenerate, as we talked about. And if that happens, so like we know people that, and this is what we do for a living, that have maybe a stroke or something and have some cell death in the brain. And we know now that neurons don't regenerate. So how do you regain function after brain damage if those neurons don't repair themselves? Well, what happens is new connections or new pathways form that can kind of reroute. So maybe like you're driving and the interstate's closed, you have to reroute and go some back roads to get to the same destination. And that's what they do. New pathways form to kind of reroute the signal and allow for function without that area being used. Synapses, these are where neurons communicate. Synapses occur between nerves from one neuron to another. They occur between the nerve and muscle, which is called the neuromuscular junction, and even between nerve and gland. So there are synapses that touch other nerves, muscles, or glands. Most of the central nervous system disorders and treatments affect the synapse. So they don't necessarily affect the cell body or the axons as frequently as they affect the synapse and what's going on there with the nerve impulses being passed along from one neuron to the next. The synapse structure. So it can be electrical or chemical. Electrical synapses send impulses quickly and are very coordinated. Chemical synapses use neurotransmitters to relay messages. So if the synapse is between a neuron and a muscle, that's called the neuromuscular junction. And then the space between two neurons is called a synaptic cleft. And within the synapse, there are synaptic vesicles which hold neurotransmitters to be used for relaying information. The cells at so like from one synapse to the next neuron, the cells in the next neuron contain receptor proteins that only work with specific neurotransmitters. So those proteins and neurotransmitters must match for the synapse to function. So if it's one specific neurotransmitter that's being released, the receptor proteins on the next neuron must match that neurotransmitter and know what to do with it in order to cause a message to be relayed. So now we're going to talk a little bit more about the function of the synapse. So an action potential at the synapse opens up calcium channels, which allows calcium to flow in and opening and open synaptic vesicles to release neurotransmitters. So an action potential at the synapse causes the calcium channels to open, allows calcium to flow in. The synaptic vessels then open to release their neurotransmitters. Neurotransmitters bind to their specific receptors on the next neuron to allow ions to flow in or out of the cell. Again, the threshold must be met for a new action potential to be stimulated, and if it's not, then the impulse will not continue. Once used, the neurotransmitter must be removed from that synapse by either being broken down or reabsorbed, which is called reuptake, so that it can be used again. So that environment has to basically be cleaned up in order to set the stage for the next one to happen. And then exocytosis is the name given for that process where the calcium channels open, the calcium is pushed, is released, and the neurotransmitter is released. That is called exocytosis. With neurotransmitters and receptors, so the neurotransmitter receptors have subtypes which determine if they stimulate or inhibit the next neuron. So each of the receptors can either stimulate a response or inhibit a response based on which part of the receptor the neurotransmitter binds to. There are large and small molecule neurotransmitters. The small molecule ones include amines and amino acids. One of the examples of that is a histamine, that is a small molecule neurotransmitter. The larger ones are peptides. An example of that is endorphins. So these neurotransmitters cause different actions based on what part of the receptor they bind to, the inhibitory or the stimulatory receptor. All right, at the neuromuscular junction, remember that is where the neuron touches the muscle to create a muscle contraction. The neurotransmitter that is used there is the acetylcholine. So acetylcholine stimulates a muscle contraction. If it can't be released or the receptors for acetylcholine are damaged, then the muscle can't contract. If um, so some diseases and drugs modify the function of the neuromuscular junction to help with allowing for muscle contractions to be created. One example of that is myasthenia gravis. That's an autoimmune disorder where the body produces antibodies to acetylcholine that destroy it and causes muscle weakness and fatigue. Um, so I have an aunt that had myasthenia gravis, and it presents a lot like multiple sclerosis. It attacks different muscle groups in the body and creates weakness there, but it actually it's those often don't recover as well as with the recovery periods of multiple sclerosis, and so then um, it can be some lasting effects as well. Making sure I said all that. All right. Oop. All right, the synaptic plasticity. So there are networks of neurons and synapses that store memories. They allow you to move. They're what detect your sensation. They are what allow thoughts, emotions, communication. So this is essentially what creates long-term memory. What allows you to learn are these networks of neurons and synapses. So new synapses are formed and old synapses are either maintained or removed based on use. Okay, so this is the basis for learning for long-term memory. Neuroplasticity is essentially synaptic plasticity. It's the rewriting of the brain's synaptic connections based on experience. So if you practice this behavior over and over and over again, your brain is rewired to do that behavior in the way that you practice it and become more and more skilled at it. If you learn a new language and then you never use it, and then suddenly years later somebody's talking to you in that language, then it's going to be a little dusty because you haven't used it. So some of those synapses are removed because you have not been stimulating those to keep them. Synaptogenesis is the creation of new synapses. So we're actually born with very few synapses, and we have to be stimulated, so sensory and motor stimulation, in order to create new synapses. So one function of PT is to help patients develop synaptic connections that increase function. There is kind of a sweet spot. I have it on this next slide, what it's called. critical period. A critical period where if you do not, I'll just stay on the next slide actually. If you do not stimulate that those synapses as they're being developed, they may never develop. So, if you think about like cases where they find children who have been severely neglected and or abused, and that maybe the child is 6, 7 years old and the people haven't been talking to them. Maybe they were like locked in a closet or just those extreme cases and they haven't been stimulated at all. They may or may not be able to develop language based on how old they are and how little language they've heard, what stimulation has, occurred there, because they might be outside of that window. So it's very important when children are developing to have a stimulating environment, to talk to the child, to touch the child, to play with them, and to allow them to move around in their environment and explore in order for all those synapses to develop and the pathways to form. Synapses that rarely activate are removed through synaptic pruning. So it's like if you don't use it, you lose it. If you're not practicing that skill, eventually it's going to be pruned away. May not be completely lost, but it will be um, it will be less present, less um, when you're able to do something really well, less well. So like an example is like riding bike. Once you learn how to ride a bike, you always know how to ride a bike. Well, if you haven't ridden a bike in 20 years and you get on, you're going to be a little wobbly, but you'd probably be able to pick it up easier than if you'd never ridden one because you did develop those synapses at some point. But some of them are not going to be as good as they were when you were a kid. All right. This is why information and skills fade with time if it's not being practiced. The continuous creation and removal of synapses is the basis for neuroplasticity. It allows for healing and recovery of function. So we're constantly making new synapses, getting rid of the ones we don't really need so much. And this is neuroplasticity. The brain is constantly changing and adapting based on the environment. There are critical periods, like we talked about, where you must stimulate an area to be organized in a usable way. So the example that the book gave is like in a baby animal, if you sew their eyelids shut, and even if nothing's wrong with their eyes or their brain, if they just don't get any visual stimulus um, for that during that critical period, then if you, after that critical period has passed, open their eyes and they're able to actually get that visual input, their brain doesn't know what to do with it. It doesn't know how to interpret it. So it would essentially cause what we call cortical blindness, meaning there's nothing wrong with the nerves or the pathways or the development of the eyes or brain, but the brain just doesn't know how to interpret the information because it's not developed the pathways to interpret that information. All right. So if you have questions, please let me know. And we can discuss those later.